Good day, esteemed students. A warm welcome to today's lecture. I am Doc Ohano, and it's a privilege to be your instructor for this course. As a registered nurse and a medical doctor, I bring both practical experience and academic expertise to our sessions. Throughout this course, we will delve into the intricate world of diagnostics with a particular focus on the fascinating realm of molecular biology and the intricate language of DNA. Together, we will explore the fundamental principles, methodologies, and applications that underpin this field, unraveling the mysteries of genetic information and its implications in healthcare and beyond. As aspiring professionals in the field of medical technology, your journey in this course is not merely about acquiring knowledge, but also about fostering critical thinking, analytical skills, and a profound understanding of the role that diagnostics play in modern healthcare. I encourage you to approach each session with curiosity, enthusiasm, and the readiness to engage in discussions and hands on activities. Together, let us embark on a journey of discovery where each lesson brings us closer to unlocking the secrets held within the strands of DNA and harnessing its power for the betterment of humanity. Once again, welcome to the class. Sir James Watson introduced the term molecular biology to describe the study of nucleic acids. While there are many molecules in nature, molecular biology focuses specifically on nucleic acids like DNA and RNA. In clinical molecular laboratories, techniques are developed to handle and analyze these nucleic acids. However, molecular techniques are now being used in other areas, such as analyzing cell surface proteins and tissue typing. Molecular biology labs can either be separate entities or part of existing units focused on molecular diagnostics or pathology. Nucleic acids, particularly DNA, have unique qualities that make them useful for clinical purposes. Techniques like hybridization and amplification allow for highly specific analyses without needing to physically or chemically select target molecules. This sensitivity enables quick and specific analyses, even with limited samples. Additionally, the sequence of nucleotides in DNA holds valuable information about normal and abnormal traits in organisms, including humans. Analyzing these sequences in the clinical lab can help predict and prevent diseases, leading to more effective treatments. DNA, or deoxyribonucleic acid, is a large molecule made up of atoms of carbon, nitrogen, oxygen, phosphorus, and hydrogen. It's like a blueprint for living things. Picture it as a long chain made up of small units called nucleotides. Each nucleotide has a sugar and the nitrogen base. That could be adenine, cytosine, guanine, or thymine. These bases are like letters in a genetic alphabet. They pair up, adenine with thymine and cytosine with guanine. These pairs are held together by bonds, forming the famous double helix structure of DNA. Back in 1871, a scientist named Masker discovered DNA, but it didn't understand its job. Then, in 1882, another scientist, Fleming, talked about the nucleus of cells but still didn't grasp DNA's purpose. Now we know that DNA, found in the cell's nucleus, stores information. This information is stored in the sequence of nucleotides, similar to how computer data is stored in sequences of zeros and ones. The sequence of these four bases, A, C, T, and G, determines all the characteristics of living things on Earth. DNA, short for deoxyribonucleic acid, has a shape like a twisted ladder, known as a double helix. This discovery was made by James Watson and Francis Crick. They based their model on earlier studies that the chemicals in DNA and physical evidence, such as X ray diffraction images taken by Rosalind Franklin, The helical shape of DNA is caused by how its building blocks, called nucleotides, arrange themselves. The order of these nucleotides on the strand and the chemicals around them can influence how the DNA helix behaves. So shown in this slide is the double helix. The phosphodiester backbones of two nucleic acid chains form the helix. Ito class yung phosphodiester bonds. Sila ang nagpo-form ng helix ng DNA. Okay? Nitrogen bases are oriented toward the center where they hydrogen bond with homologous bases to stabilize the structure. And ito yung mga base pairs. Diba? A is to T, G is to C. Next slide. So what are nucleotides? Nucleotides are about 700 kilodaltons in size. Each nucleotide comprises a 5-carbon sugar molecule with a nitrogen base attached to the first carbon and a triphosphate group linked to a 5th carbon. So what are nucleosides? When a nitrogen base is attached to an unphosphorylated sugar, it forms a nucleoside. Common nucleosides include adenosine, guanosine, cytidine, and thymidine. If the ribose sugar is phosphorylated, it becomes a nucleotide. No. Kanina class, nakita nyo may yung nucleotide, may nakabit na phosphorus molecule dito, na phosphate, sa sugar. Sa nucleoside, walang nakakabit na phosphate sa sugar. But, ano lang siya, nitrogenous base plus sugar in the form of a ribose. The 5-carbon sugar found in DNA is called deoxyribose, which is a modified form of ribose with a hydrogen atom instead of a hydroxyl group attached to the second carbon. The hydroxyl group in the third carbon is crucial for forming the backbone of DNA strand through phosphodiester bonds. Nitrogen bases are flat carbon-nitrogen ring structures. DNA has 4 common nitrogen bases, namely adenine, guanine, cytosine, and thymine. These bases are distinguished by variations in substitutions and bonds within their ring structures. Thymine and cytosine, which have a single ring, are called pyrimidines, while guanine and adenine, which have a double ring, are called purines. So, position numbering in nucleotides starts with the nitrogen base ring positions, designated as C or N, 1, 2, 3, etc. The carbons of the ribose sugar are numbered 1 to 5, distinguishing them from the nitrogen base rings. The nitrogen bases in nucleotides form specific hydrogen bonds with each other. Guanine pairs with cytosine through 3 hydrogen bonds, while adenine pairs with thymine through 2 hydrogen bonds. These hydrogen bonds are crucial for maintaining the specificity of DNA-based tests and preserving the information encoded in the nucleotide sequence. During DNA replication, new nucleotides form hydrogen bonds with complementary nucleotides on the parental DNA strand, A with T, G with C, allowing for accurate duplication of the nucleotide sequence. Mismatches or base pairs other than T, and GC can distort the DNA helix and disrupt sequence information maintenance. So what are substituted nucleotides? Nature often alters the structure of nucleotides in various ways. These alterations include adding methyl groups, removing amino groups, adding new chemical groups, substituting parts of nucleotide, and other modifications. These changes create nucleotides with different characteristics. For instance, adding a methyl group to certain nitrogen bases can affect how genes work in living organisms, and this is a natural process. However, external factors like exposure to chemicals or radiation can also cause these changes, which may lead to negative effects like cancer. So, Modified nucleosides, because of their impact on enzymes that handle DNA, have proven useful in medical treatments. So these are some substituted nucleosides used in the clinic and the laboratory: deoxynucleoside, dideoxynucleoside, azidothymidine, and acyclovir. Some drugs used to fight cancer, like 5-bromouridine and cytosine arabinoside, are altered forms of thymidine and cytosine nucleosides respectively. Other drugs, such as azidothymidine, 2,3-dideoxy-2-fluoro, and 2,3-dideoxyinosine, are used to treat patients with HIV and are modifications of thymidine, cytosine, and adenine, respectively. Acyclovir, which is a medication used against herpes simplex virus and varicella zoster virus, is a modified version of guanosine. In research laboratories, scientists can alter nucleosides for purposes like labeling or detecting DNA molecules, sequencing, and other experiments. The methods for these procedures will be explained in later chapters. Now, let's move on to nucleic acid. Nucleic acids are big molecules made up of building blocks called nucleotides. These nucleotides stick together through their phosphate and hydroxyl groups. When a nucleotide joins a chain, it hooks onto the last nucleotide's hydroxyl group using its phosphate group. This process of adding nucleotides creates a directionality to the DNA chain. Imagine it like a road with a start and with an end. The DNA chain has a start with a phosphate group and an end with a hydroxyl group. Scientists call this directionality 5' to 3', meaning the nucleotides are read in that order along the chain. In nature, DNA usually comes in pairs, forming a double-stranded structure. These strands run in opposite directions, kind of like two lanes of traffic going in opposite directions on a highway. They're held together by hydrogen bonds between specific pairs of bases, namely adenine with thymine and guanine with cytosine. The bases are arranged in a spiral around each other, like a twisted ladder. Each strand of the DNA double helix holds a copy of the genetic information. The sequences of two strands are complementary but not identical. They run in opposite directions, with one strand starting where the other ends. This setup ensures that identical sequences don't stick together. This is important for designing experiments that involve combining DNA strands, like in genetic testing or research. Now, just a recap of your DNA replication class. This is the process of making a copy of DNA molecules. DNA strands are oriented in opposite directions, which is important for how they're copied. The copying process occurs from 5' to 3' direction, guided by DNA polymerase, which is the enzyme that adds nucleotides based on a template. This enzyme reads the template in the 3' to 5' direction. This results in a double-stranded DNA molecule with one parent strand and one newly synthesized strand, but both oriented differently. Okay? This illustration class shows us the simultaneous replication of both strands of the double helix. Both strands are read in the 3' to 5' direction. The lagging strand is read discontinuously. Ito class yung lagging strand. With the polymerase skipping ahead, ito yung polymerase. Another one here. Skipping ahead and reading back toward the replication fork on the lagging strand. Yan sila class. So, semi-conservative replication, as predicted by Watson and Crick, is vital for passing on genetic information accurately to new cells. This process ensures that sequence of nucleotides remains the same across generations. During replication, each strand of the original DNA serves as a template for building a new complementary strand. The process starts with unwinding of the DNA double helix, allowing each strand to act as a template. New nucleotides are added to the template strand, forming H bonds and then bonded together, creating a new DNA strand. Replication proceeds through both DNA strands in one go. Electron microscopy shows this as a forked structure called a replication fork. However, copying both strands simultaneously in the same direction is not possible due to the anti-parallel nature of DNA. To solve this, Okazaki and Okazaki discovered short DNA fragments called Okazaki fragments, formed during replication. The lagging strand is copied discontinuously in small fragments, while the leading strand is copied continuously. Primase, an enzyme, lays down short RNA primers on the lagging strand, which are needed for DNA synthesis to begin. Polymerases. DNA polymerases are enzymes that play a crucial role in copying DNA. In prokaryotes, organisms like bacteria without a cell nucleus, the first enzyme discovered to do this job was called DNA polymerase 1 or POL1. Later, two more polymerases were found, namely DNA polymerase 2 and polymerase 3. Among these, DNA polymerase 3 is the primary enzyme responsible for copying DNA during bacterial replication. Polymerase 1 was the first to be studied in detail, likely because it was easier to isolate at that time. Polymerase 2 and 3 were found to be less active in early lab tests compared to polymerase 1. In living organisms, polymerase 3 works as part of a larger team of proteins needed for DNA replication. Polymerase 3 is made up of several subunits, two of which are the actual enzymes responsible for adding new DNA building blocks during replication, one for each strand of DNA being copied. DNA polymerase enzymes not only build new DNA strands, but can also remove or replace incorrect DNA building blocks. This proofreading function helps ensure accurate copying of DNA sequences. One part of the DNA polymerase enzyme, called the cleanout fragment, is often used in laboratory experiments because it can replicate DNA without proofreading. The exonuclease function of DNA polymerase helps maintain the accuracy of DNA copying by removing mismatched nucleotides before adding new ones. During replication, DNA polymerase 3 can add and remove DNA simultaneously at the nick in one strand of the DNA. This process, called nick translation, can be useful in labeling DNA for experiments. Another type of DNA polymerase called terminal transferase can add new DNA building blocks without needing a template. This enzyme is used in the lab to label DNA. DNA polymerases are essential in biotechnology for tasks like cloning, amplifying DNA, and sequencing. Different types of polymerases have been identified and categorized into families based on their structure and function. The A and B families of DNA polymerases are particularly useful in biotechnology because they are made up of single proteins which can be modified to have specific characteristics needed for different experiments. These modifications can include changes in how long the enzyme stays attached to the DNA template, how accurately it copies the template, and its preference for certain DNA building blocks. So, what are the enzymes that metabolize DNA? After DNA is formed, it's not just sitting there doing nothing. The information it holds needs to be used selectively to create RNA while also being protected from mistakes or mutations. Plus, when organisms reproduce, it's important to mix up the genetic information to create diversity in the offspring. This mixing involves cutting and rearranging DNA strands before cells divide and gametes, sperm and egg cells, are formed. There are many enzymes involved in these processes throughout the cell cycle. Some of these enzymes, like DNA polymerase, have been isolated and can be manipulated in the lab outside of living organisms. They're really important for techniques like recombinant DNA technology, which is widely used in molecular biology. First, enzyme restriction enzymes. These are like molecular scissors found in bacteria. They're called endonucleases because they cut DNA internally, breaking the backbone of the DNA molecule. These enzymes are picky and only cut DNA at specific sequences of bases. Originally, scientists discovered restriction enzymes in bacteria where they act as a defense system against foreign DNA. These enzymes recognize specific sequences in the DNA and chop it up, limiting the ability of foreign DNA to harm the bacterial cell. The bacteria can tell the difference between their own DNA and foreign DNA because of a process called methylation. Each restriction enzyme is named after the bacterium it comes from. For example, there's one called BAMHI from Bacillus amyloliquefaciens H, and another called HIND3 from Haemophilus influenza. Restriction enzymes. So we have three types of restriction enzymes. Type 1 are enzymes that have both cutting and methylation activities. They recognize specific DNA sequences and can cut the DNA strand far away from the recognition site. Type 2 restriction enzyme is commonly used in laboratories. They recognize specific DNA sequences and cut the DNA directly at those points. They create predictable fragments with sticky ends that can easily join with other DNA fragments. Type 3, similar to type 1 enzymes, they can both methylate and cut DNA. They recognize specific DNA sequences and cut the DNA a certain distance away from the recognition site. DNA ligase. It is a protein that helps stick pieces of DNA together. It forms a bond between the ends of DNA molecules where there's a 3-hydroxyl and 5-phosphoryl group. Scientists figured out it existed because they saw DNA copying, mixing, and fixing happening in living things. And they knew something had to be joining the DNA pieces together. DNA ligase was found in 1967 by researchers in 5 different laboratories. They found that this enzyme could connect cut pieces of DNA back together. So, other nucleases, exonucleases. In contrast to endonucleases, which cut DNA in the middle, exonucleases break down DNA starting from the free 3' hydroxyl or 5' phosphate ends. This means they won't work on DNA that's in a closed circular form. Exonucleases are used in controlled laboratory conditions to manipulate DNA in specific ways. For example, they can be used to make gradual deletions in linear DNA or to modify DNA ends after cutting them with restriction enzymes. Different exonucleases have different preferences for the types of DNA ends they degrade. Exonuclease 1 from E. coli breaks down single-stranded DNA starting from the 3' hydroxyl end, creating individual nucleotides. It works best on long stretches of single stranded DNA and slows down as it nears a double stranded region. Exonuclease 3 from E. coli removes individual nucleotides from the 3' end of double stranded DNA in the presence of certain metal ions. It also has some ability to cut DNA in the middle at specific sites. Exonuclease 3 can work on various types of DNA ends but won't digest 3' overhangs. In research, it's used to create specific deletions in double-stranded DNA or to generate single-stranded DNA for certain sequencing methods. Exonuclease 7 from E. coli breaks down single-stranded DNA from either the 5' phosphate or 3' hydroxyl end. It's one of the few enzymes that can work from the 5' end. Exonuclease 7 can be used to trim long single-stranded sections protruding from double-stranded DNA. There's a type of enzyme called nuclease BAL31, which comes from a bacteria called Alteromonas espesiani. This enzyme can break down both single-stranded and double-stranded DNA from both ends. It works slowly at 20 degrees Celsius, which means it can be controlled precisely, making it really useful for scientific research. Scientists often use it to remove specific sections of DNA, a process known as making nested deletions. Class, ano tong mung bean? Ito yung munggo. Well, guess what? May nuclease na derived from mung bean. Mung bean nuclease is an enzyme derived from mung bean sprouts. It breaks down single-stranded DNA and RNA molecules. What's unique about it is that it doesn't touch double-stranded DNA regions. Scientists use it to tidy up the ends of DNA pieces that were cut by certain enzymes. This cleanup process helps create blunt ends, which are useful for genetic cloning experiments. S1 nuclease. S1 nuclease is an enzyme derived from a fungus called Aspergillus oryzae. This enzyme specifically targets single-stranded of DNA or RNA, breaking them down into smaller units called mononucleotides. Additionally, it can cut within single-stranded areas of double-stranded DNA like gaps or loops. Scientists used S1 nuclease a lot in the past for RNase protection assays, which are experiments to study gene expression. It's also handy for a technique called nuclease mapping. Direct BCM enzyme, found in E. coli bacteria, is a type of enzyme that works on DNA. It needs ATP, your adenosine triphosphate, to do its job, so it's energy requiring. This enzyme can cut both single-stranded and double-stranded DNA molecules. However, it doesn't work on small gaps in the DNA called nicks. It can cut DNA from either end, either from the 3' hydroxyl or from the 5' phosphate end. When it cuts double-stranded DNA, it creates shorter fragments called oligonucleotides. Micrococcal nuclease is an enzyme that breaks down DNA and RNA molecules. It's particularly good at breaking down DNA and RNA that have a lot of A and T bases, or A and U bases in RNA. While it can break down both single-stranded and double-stranded DNA, it prefers working on single-stranded DNA. In laboratories, scientists use micrococcal nuclease to clean up DNA and RNA from messy mixtures, and also to study how DNA is organized within cells. DNase 1 or deoxyribonuclease 1, it is an enzyme found in the pancreas of cows. Its job is to break down DNA molecules into smaller pieces called oligodeoxyribonucleotides. It mainly works on DNA strands that have pyrimidines in them. Technically, it's called an endonuclease because it breaks down the DNA from within. Scientists use DNase 1 in research labs and medical labs to separate DNA from RNA samples. It's also used to find parts of DNA that are exposed in experiments where DNA interacts with proteins. DNA polymerase 1. In E. coli bacteria, there's an enzyme called DNA polymerase 1. This enzyme has a special ability known as exonuclease activity. Before, it used to be called exonuclease 2. This activity helps the enzyme in a process called proofreading, which means it can correct mistakes as it builds strands of DNA. Nucleases are enzymes found naturally in cells. When preparing DNA for medical tests, it's crucial to remove or deactivate these enzymes. Most methods for isolating DNA are created to reduce the activity of both types of nucleases, whether it be endonuclease or exonuclease. The purified DNA is typically stored in a solution called PE buffer, which contains tris hydrochloric and EDTA. This buffer helps prevent nucleases from working by binding to the metal ions they need to function. Helicases are enzymes that play a crucial role in managing the structure of DNA, which doesn't just exist as a simple double helix, but rather in organized loops and coils. They help in tasks like transcription, replication, and combination by cutting and rejoining the sugar phosphate backbone of DNA. This prevents tangling and allows the DNA to function properly. When DNA needs to be accessed for processes like replication or transcription, it can become tightly wound or tangled. Helicases step in to relieve this stress by cutting and rejoining the DNA strands, allowing them to unwind and separate as needed. There are two main types of helicases, namely topoisomerases and gyrases. Topoisomerases help to relax the tightly wound DNA by changing its structure, while gyrases, a type of topoisomerase, specifically untangle DNA by creating double strand breaks and separating linked rings of DNA molecules. In eukaryotic cells, like those in plants and animals, topoisomerases function similarly to those in bacteria, but with some differences in their mechanisms. These enzymes are critically important for cell replication, making them targets for certain anti-cancer drugs. These drugs work by interfering with the actions of topoisomerases, ultimately leading to cell death by disrupting the DNA cutting and joining processes. Methyltransferases are enzymes that add methyl groups to certain parts of DNA molecules. In simpler terms, they modify DNA by attaching a small chemical groups called methyl groups onto specific spots. In prokaryotic cells like bacteria, DNA is often fully methylated or partially methylated, meaning only one side of the DNA strand gets methyl groups. This helps the cell identify its own DNA and protects it from certain enzymes. On the other hand, in more complex eukaryotic cells like in plants and animals, DNA methylation happens in specific regions. Proteins in the cell can control where this methylation occurs. Now, there are two main types of methyltransferases. One is maintenance methyltransferases. These enzymes work continuously throughout the cell's life cycle and add methyl groups to DNA that is partially methylated. On the other hand, de novo methyltransferases are active mainly during early development, during embryonal development. and are involved in creating specific methylation patterns in different types of cells. So recombination in sexually reproducing organisms. Recombination is when new combinations of genetic material are created. This happens through a process called crossing over, where molecules exchange parts of their DNA. When an organism or molecule has a new mix of DNA sequences, it's called recombinant. In sexually reproducing organisms, each new generation has a mix of genetic material from the parents. This mixing increases genetic diversity, which can lead to offspring that are more adaptable and healthier. This is seen in hybrid individuals, which often show stronger traits compared to purebred organisms. There are three main ways genes mix in sexually reproducing organisms. First, during meiosis, chromosomes exchange genetic material through crossing over. This creates new combinations of genes on the chromosomes. The newly combined chromosomes are then randomly sorted into gametes. When two gametes from different parents merge during fertilization, they bring together their own unique sets of recombined chromosomes, resulting in offspring with a mix of genes from both parents. So, this illustration, generation of genetic diversity by crossing over of homologous chromosomes. This mixing of genes leads to the inheritance of traits in subsequent generations. Recombinant DNA technology is a method of controlling gene mixing. Instead of relying on natural processes, scientists can manipulate single genes by altering, replacing, deleting, or moving them into new genomes. This targeted approach allows for the creation of organisms with specific traits, similar to purebred organisms, but with individual gene differences. This technology has implications not only in research laboratories, but also in the treatment and prevention of diseases, such as through gene therapy. Recombination in asexual reproduction. Movement and manipulation of genes in the laboratory began with observations of natural recombination in a sexually reproducing bacteria. Genetic information in asexually reproducing organisms can be combined in three ways, namely conjugation, transduction, and transformation. These are all processes by which bacteria transfer genetic material, but they operate differently. Conjugation involves two types of bacteria termed F plus and F minus. For conjugation to happen, F minus and F plus cells need to be in contact. The F positive bacteria carry a fertility factor that facilitates this contact and transfer genetic information from the F plus to F minus bacteria. This transfer can occur through a bridge-like structure between the bacteria. The F plus factor, or the fertility factor, which is a circular DNA molecule, can also integrate into the host chromosome, creating HFR bacteria. These HFR bacteria can transfer both the F factor and parts of the host chromosome to F bacteria. High frequency. Um, this figure illustrates the conjugating cells must be in physical contact with each other. Ito class, naghalo-halo sila. Physical contact with each other to successfully transfer the F positive phenotype. If the cells are separated by a membrane, in this case itong nasa baba, the F minus bacteria, itong blue, do not become F positive. No? So they should come in contact first for asexual reproduction to be successful. Next slide. Fertility, the ability to donate genetic information, is controlled by the F factor, which is shown here by the color green circle. The F factor can exist by itself or can be reintegrated into the host chromosome. Tingnan nyo class nangyari dito na integrate siya sa chromosome ng host, or pwede siyang standalone dito. Now, transduction is a process where genetic material is transferred from one bacterium to another by bacteriophages. These are viruses that infect bacteria. The viral DNA enters the bacterial cell carrying genetic information, while the viral protein remains outside. This process was confirmed by Hershey and Chase using labeled DNA and protein. Class, this is a sample of a bacteriophage. Bacteriophage infects a specific strains of E. coli. It has specialized structures. The tail fibers find the bacterial surface and allow contact of the tail plate and injection of the DNA in the viral head through the sheath into the bacterium. Ito class yung DNA. Tapos i-inject nila yan sa bacteria through this sheath. Okay? That's how they infect bacteria. Transformation was first observed by Griffith. This involves the direct uptake of DNA by bacteria. Griffith noticed that when he killed smooth type bacteria with live rough type bacteria, the rough type became smooth and virulent. So, recall in our history of molecular biology topic, we discussed the transforming factor discovered by Griffith was responsible for changing the phenotype of the avirulent rough type bacteria to that of the virulent smooth type. Avery, MacLeod, and McCarthy later identified DNA as a transforming material by demonstrating that treatments degrading proteins and RNA didn't affect transformation, while degradation of DNA did. So, Avery, MacLeod, and McCarthy showed that destruction of protein or RNA in the cell lysate did not affect the transforming factor. Only destruction of DNA prevented transformation. Ito class, dahil nalusaw niya yung DNA, that's why na-prevent yung transformation. Now, in summary, conjugation involves direct cell-to-cell contact and transfer of genetic material via a bridge. Transduction uses viruses to transfer DNA between bacteria, and transformation is the direct uptake of DNA from the environment by bacteria. These processes were crucial in understanding bacterial genetics and laid the foundation for modern genetic engineering techniques. Plasmids. Plasmids are small circular strands of DNA found in cells, especially in bacteria. Unlike the linear DNA found in most organisms, bacterial chromosomes are often circular. Plasmids are like extra bits of genetic information that carry bacteria or that bacteria can carry alongside their main chromosomes. Plasmids are small extrachromosomal DNA duplexes that carry genetic information. Ito yung circular plasmid class. Tapos, those plasmids are composed of, usually mga genes necessary for DNA transfer, and then antibiotic resistance genes, versus ito yung main circular chromosome ng bacteria. Okay? So, they're usually around 2,000 to 100,000 base pairs long. Just like chromosomes, plasmids contain genetic instructions. However, because of their small size, they can only carry a limited amount of information. So, supercoiled plasmids can be relaxed by nicking Uh, by local unwinding of the double helix. Kita nyo klase itong supercoiled structure ng DNA ng plasmid. Through the help of these enzymes, gyrase plus adenosine triphosphate, marerelax niya yung coil. And conversely, para mag-coil uli yung relaxed circle, you will be needing this enzyme, this topoisomerase. Plasmids can exist in different forms. They can be supercoiled, twisted, or linear, depending on whether their DNA strands are broken. Scientists can study these different forms using a technique called gel electrophoresis. Plasmids were found to be important because they can carry genes that make bacteria resistant to antibiotics. This was first discovered when certain dyes could reverse antibiotic resistance in bacteria by affecting plasmids. These plasmids carrying resistance genes were named resistance transfer factors or R factors. They often carry resistance to antibiotics like chloramphenicol, tetracycline, and others. Plasmids can replicate inside a cell but can't survive outside of it, like viruses. They are thought to acquire resistance genes from other bacteria. These genes can be gained or lost by plasmids in the bacterial population. Plasmids can also exchange genes with each other, leading to new combinations of resistance traits. There are two main types of plasmids, large and small. Large plasmids like the F factor carry their own instructions for transfer between cells. They're present in smaller numbers in cells. Small plasmids, on the other hand, are more common but don't have instructions for their own maintenance. They rely on being copied and distributed when cells divide. Alright, so end of lecture class.